Welcome back, everyone. This lesson will cover what neurons are and how electrical signals are transmitted. Our brain contains numerous neural networks. The smallest unit of these networks is the neuron. Neurons are specialized cells that can transmit electrical signals to various body parts. They have four main parts: a cell body, dendrites, and axon terminals. The cell body, also called the soma, contains the nucleus and most of the neuron's cytoplasm. This is where proteins are formed and transported to make sure the cell functions properly. Dendrites are these branched projections that collect incoming signals from other neurons. Neurons pass signals by sending electrical impulses along its axon, which is this long extension that ends in axon terminals. At these axon terminals, the signal is passed across a very narrow gap to the dendrites of other neurons. This gap is known as a synapse or synaptic cleft. Now let's further examine how the neuron relays its signal along the axon. The neuron's cell membrane is filled with ion channels, which are proteins that act like tunnels that only allow certain ions to enter or leave the cell at certain times. This is influenced by the difference in potential charges across the membrane, or the membrane potential, also known as the voltage. When a neuron is not firing and in resting state, the membrane potential is around negative 70 millivolts. This means the inside of the cell has fewer positive ions than the outside, so the overall more negative. Now, when the neuron receives an excitatory signal at its dendrites from other neurons, this triggers ion channels to open, allowing positive ions from the outside to enter the cell. This makes the membrane potential less negative. Now, if the sum of all the signals at the dendrites rises above a certain threshold, more ion channels nearby will open automatically, causing even more ions to move across the cell membrane. At its peak, the membrane potential increases to around positive 40 millivolts. This process is called depolarization. Ultimately, this creates an electrical impulse called the action potential, and it moves down the axon towards the axon terminals. Interestingly, the axon is wrapped in fatty substances called myelin sheaths. This helps to insulate the action potential so it gets transmitted faster, similar to why electrical cables have plastic insulators. To increase the speed of the action potential, high concentrations of ion channels are present in the parts of the axons that are not myelinated, known as the nodes of Ranvier. This allows the action potential to essentially jump from node to node really quickly, a process called saltatory conduction. Now, when an action potential arrives at the axon terminal, the change in membrane potential triggers a specific set of ion channels to open. As a result, calcium ions flow into the cell, and they bind to vesicles or packages that contain signaling molecules called neurotransmitters. These vesicles then move and fuse with the cell membrane at the axon, ultimately emptying their contents into the synapse. The neurotransmitters then bind to receptor proteins found on the dendrites of other neurons. The cycle repeats, and the action potential is passed on to the next neuron. To clarify things, we call the neuron releasing neurotransmitters the presynaptic neuron, and the neuron receiving the neurotransmitters the postsynaptic neuron. A postsynaptic neuron has two general types of receptors on its dendrites. What we covered just now, where the neurotransmitter binds to the receptor and the ion channels open, are called ionotropic receptors. The other one is called metabotropic receptors, which are more complex. When the neurotransmitter binds to the metabotropic receptor, it triggers a cascade of biochemical steps. An ion channel that is further away may open, or some other molecule may get activated, for example. So then what happens to the neurotransmitters after they trigger an action potential? In some cases, the enzymes in the synapse break down the neurotransmitters so they don't continue triggering the neuron. Other times, they are reabsorbed in a process called reuptake. Finally, neurotransmitters can be excitatory or inhibitory. This means they can either pass on an action potential or prevent it from reaching the next neuron. The brain's most common excitatory neurotransmitter is glutamate, and the most common inhibitory neurotransmitter is gamma-aminobutyric acid, or GABA. That's it for now. Thanks for watching and stay tuned for the next episode, where we touch on the roles of glial cells in the nervous system.